And now is Sergio Díez Hermano with machine learning presentation of loss of eye regularity in a Drosophila neurodegenerative model. So,
okay. Can I use the mouse? Yeah. Okay, let's see. I can break anything because I think we will. Oops. So, okay. Let's see if I can put this laser here. Okay, nice. Maybe I'm looking at the monitor so I don't have to be turning around all the time. So, good evening, everyone. Thanks to the organizing committee for having me here today. First of all, I think I'll do some presentations. My name is Sergio. I've recently joined the forest pathology group here in Valencia in a postdoc position, and my background consists essentially in data analysis and biostatistics. So for today, I'm going to explain some data analysis techniques which are called machine learning as a term. And in particular, I'm going to show you the biological application to some real data, which is a case study in a Drosophila melanogaster neurodegenerative model. Okay, so a little bit of biology. As you can see in the slides, maybe this talk is a little bit farther from the main topic of this symposium, which was sustainable management of forests, but I hope you will find at least the data analysis part transferable to your research projects. So what you can see in the slide is the visual system of the fruit fly, which is the common name for Drosophila melanogaster. Fruit flies have compound eyes, which are made of small, tiny units, which are highly organized. They are organized in a particular spatial arrangement, which is very regular. They have even a geometric arrangement. And the interesting thing is that we can disrupt this regularity. We can degenerate the geometry to study human diseases. One of these diseases is the spinocerebellar ataxia. This is a genetically inherited disease, which basically is related to loss of mass of gray matter in the cerebellum of the patients. So in the end, they have motor impairment, they have weak fine motor skills, they lack coordination, and it is a neurodegenerative disease. So the symptoms get worse with age. And we can use fruit fly to study if there is any way to find out new treatments to this disease. So one thing we can do is to create transgenic lines of fruit fly which are overexpressing particular genes which are known to be behind this neurodegenerative disease. And we can not only overexpress the genes responsible for the human disease in the fruit fly, we can do it in a specific tissue. In this case, in the retinal surface of the eye. So what you can see at the bottom of the slide is the healthy control eye, absolutely regular and geometrical. And you can see here are two examples of fruit fly eyes which are overexpressing the human disease gene. And as you can see, the retinal surface is completely disrupted. It is degenerated. And in the close-ups, you can see that there is absolute loss of regularity. The truth is that these really detailed pictures that come from electron microscopy, many laboratories around the world cannot afford to gather really big samples of this kind of pictures. So they end up using pictures like the ones you see here, which are bright field pictures. They are not as detailed and they are harder to quantify. Another question is, how do I quantify what I see here? What is the percentage of regularity I see in the wild type, in the full healthy eye, in comparison to the fully degenerated eye? How do I do this? And even more, what if I have an experimental setup that consists on combining in the same fruit fly the genes responsible for the disease and some genes that are known to be neuroprotective? So now I want to measure whether these neuroprotective genes have some capability to restore the original phenotype, or at least what is the degree of rescue that they can offer. So maybe we can go further in humans and study these genes if there are some interesting traits. So these pictures we see here at the bottom. Is this 80% the original phenotype? Am I rescuing the original phenotype in a 50, 60%? What is the number? How do I quantify the rescuing of the original phenotype with my neuroprotective genes? The answer to this question is machine learning. Okay, let's see here. Here what you can see are two different approaches to the analysis of this kind of data with machine learning. At the top, we see what we call the classical approach in machine learning, which is called classical because these algorithms cannot understand the original picture as it is. You have to translate the picture. You have to extract the most interesting features of the picture so the algorithms know what is the information contained within the images. In contrast, you have the modern deep learning, which is the gold standard, which consists in neural networks. Neural networks are called this way because they mimic the networks in the human visual system, and they are particularly well suited to understand pictures as they are. They don't need a translation step. You get the pictures, and the deep learning algorithm can automatically understand what is going on in the picture because they have these consecutive layers of processing. So in the end, you end up comparing which is the best machine learning algorithm that I can use to quantify my data and to correctly classify my images as a wild type, as a degenerated, or as an intermediate phenotype. The first thing you want to do is always an automatic selection step because I need to extract the region of interest from my pictures. And this has to be automatic to avoid any human intervention, to avoid any bias caused by subjective human decisions. Even more, if you have a bias that comes from an automatic procedure, you can measure the bias, you can reproduce the bias, and you can communicate what is the problem that is going on there. And as you can see here, the pipeline is more or less intuitive. You have the original pictures, which has parts of the fruit fly that you are not interested in initially. You have the upper part of the body, you have other parts of the head, you have even the forelegs there. You don't want that, you don't want to quantify that. You want to extract the pixels that correspond to the eye, to the retinal surface. And to do that, the algorithm automatically starts selecting candidates, pixels that might belong to that region of interest. They start doing clustering and saying, okay, this pixel has more probability than this one to belong to the region you are interested in. And in the end, you have a data set like this one. Here you have approximately 30 pictures, which consist essentially in regions of interest extracted from the original pictures. And as you can see, sometimes the algorithm works almost flawlessly, but sometimes the algorithm doesn't work that well, and most probably because of the source material is not always of the best quality, and the algorithm can only work as better as your source material is. So this is the data set you have. And then comes the funny part, which is training your machine learning algorithm. You select random samples of your dataset, and you train your model to learn which are the particularities of your dataset that they can use to predict new unseen pictures they've never been exposed to, because you are interested in the generalization capabilities of your algorithm. Can my algorithm correctly predict something it has never seen before? And that's what you see here in this the representation. For example, here we have 4 different classical machine learning algorithms and comparing their performance. And what you want is to have a perfect diagonal, so every new picture is correctly classified in the phenotype they belong to. And as you can see in this example, the support vector machine, that is a particular algorithm, was the best candidate with almost perfect performance. Now you have to compare whether this classical machine learning algorithm is better or worse than the deep learning, the neural networks, which is the gold standard. And here you see a representation of the internal structure. These consecutive layers are what allow the neural network to understand the picture. They know what is a border, what is an edge, whether there is much contrast or not in the picture, and then you input your own data set at the end, do a little bit of fine-tuning, and then you get the results and say, okay, my deep learning algorithm is almost perfect in terms of accuracy performance, but the improvement I got in comparison to the classical is really tiny. I got a 0.01 increase in performance, and we have to take into account that neural networks are much more expensive computationally than classical machine learning. So when you talk to another researcher who wants to use your algorithm, you have to say, okay, is it worth it, the trade-off? Because your computer has to be much, much more powerful to run these kind of algorithms. And in the end, for every picture, you got the number, you can plot distributions for your phenotypes, and on this kind of data, you can use any kind of statistics, p-values, and whatever you want to use to say to other researchers, okay, there are significant differences in my data set. And as a final remarks, the conclusion you have from this kind of algorithms can be made extensible to many other scenarios, whether it be genomics data, prediction, or practically any kind of data set. You always need an automatic first step of selection of your regions of interest. Then you need to compare different machine learning algorithms to see what is the best one that best fits your data. And in the end, as a developer, you are always interested in developing user-friendly interfaces, web application, because at the end, your ultimate goal is to reach out to other researchers. They have to be able to use your algorithms to do their own parameter tweaking, to train their new models, and eventually you get a new citation from your work, which is always nice. And that's it. Thank you for being here today, and I'll gladly answer any question. Okay. Next.